In this video, we are going to learn how abundance is measured. Measuring the different levels of biodiversity within an ecosystem can be a tasking job. Finding out which species live in an ecosystem and the size of the population requires the identification and the cataloging of all organisms present to build up a species list. This can be possible for areas that are very small or where the species are very large, like the trees. However, for larger and more complex ecosystems like the rainforests, it is simply impossible to find, identify, and count every organism that exists there. When this is the case, different samples of the area can be taken and used to make an estimate for the total species numbers in the area. And one way to do so is by sampling. So, sampling is a method of investigating the abundance and distribution of species and population. And we have two different types of sampling. We have the random sampling using the frame quadrats or point quadrats, and we have the systematic sampling along a belt transect. So what is the difference between random and systematic sampling? Well, in random sampling, the positions of the sampling points are completely random or due to chance. So, this means that this method is beneficial because it means that there will be no bias by the person that is carrying out the sampling that may affect the results. On the other hand, in systematic sampling, the positions of the sampling points are chosen by the person carrying out the sampling. This means that there is a possibility that the person choosing could show bias toward or against certain areas. And this could be unrepresentative of the whole area. And in the next video, we will elaborate more about quadrats and transects.